Okay, good day, everyone, and I welcome you to this class on entrepreneurship, which we are going to be tailoring towards open labware. And because this is an entrepreneurship class that has to do with historical materials, so this is going to be an eye-opener about the fact that there is entrepreneurship in open labware beyond just the histological material, right? So we're going to be citing a couple of examples and some illustrations that makes you appreciate the potential, the entrepreneurial potential in labwares generally, or things that have to do laboratories. So to proceed, in this case, we nice that we appreciate the convention. And the convention is not too far from us all. And that includes the fact that in histology, if you are a histologist or you're somebody that is into histology, the conventional ways to earn some income includes tissue processing, right? Or tissue staining, just generating some samples that can be viewed as a result of some research work that has been conducted in vivo would take different stages, right, of processing. So that includes the tissue processing. It is something that would earn one money on its own. Tissue staining, it also has its entrepreneurial potential. Then photomicrographing, that is to take the photomicrograph. It's something that is also, it also has its potential. Then to add statistical analysis to this, so to convert in a qualitative result that is photomicrographs are qualitative results. They are qualitative data. Then you can, in order to convert them into quantitative data, you start having to count cells to be able to quantify some things, or you start quantifying the level of staining, the staining intensity. So this makes the data sort of semi-quantitative, even though it's not essentially quantitative in its start. So this is also something that is of its own potential. Then interpretation of the slides to explain what goes on within the tissue and the implication of the result is also something. So these are the conventions generally. So other straightforward conventions will be dealership in histological materials or reagents or equipment. These are things that we have known as the convention, right? Now, looking at the materials, if you look at these different equipment, laboratory equipment, you would agree that they amount to a lot and they also indicate how much of them we remain needy of on the continent at large, not only in the country. So it is agreeable that we have a whole lot of needs on our continent in terms of equipment and tools that would be used for research. But come to think of it, if you look at the word population, you can appreciate how many percentage of the entire world population is constituted by Africa, right? This is looking at how many people reside in each continent. This is a substantial representation of African population in the world, right? But if you wish to look at this population map from another lens, like how many of these inhabitants of the whole world population, where do we have the concentration of Nobel laureates, which is like the hallmark of research accomplishment, right? We see a complete change in the density of this population map, such that Africa is hardly visible somewhere around here. So you can appreciate that the bulk of those Nobel prizes and Nobel laureates kind of come from the global north, what we call the global north, right? The Northern America, Europe, and some part of Asian countries. So this changes the outlook of the map entirely. And that is not surprising because it seems directly proportional to how much of scientific papers are coming out from this continent. So if you look also through the scientific lens, the lens of scientific papers, you would realize that Africa is also not well represented, right? Almost shrinking here. We still have the global north taking the stage. So the question is, how do we become, or how does Africa become the like of the global north? What is the way forward? If you want to produce similar amount of science, you want to achieve the similar accomplishment in terms of research, what is the first thing that comes to mind in being able to meet up or match up with the global north? Money becomes the first issue, right? And we know that we haven't got the money, and therefore we'll have to think, as in the words of Rutherford. So it begs the question of, if there is any alternative means of achieving some of these things, alternative means of making the global south, which includes Africa, match up or meet up with the kind of accomplishments that are obtainable in the global north. So one of the essential tools that we'll discuss today is 3D printing and its relevance. And suffice to say that, because this is not a dedicated class on that, on 3D printing, suffice to say that there are 7 main categories of 3D printers, right? It's not just one. There are 7 main categories of them, not just one. So, because they run on different principles. So one of them is VAT polymerization, another is material extrusion. So the material extrusion is the most common principle and is usually the recommended one for beginners. So this is where you have molten thermoplastic deposited through a heated nozzle. Such as you see on this picture, this is a typical 3D printer, and we'll talk in a minute about how it runs and the principle. So this is the FDM filament deposition model system. And this one you see on the right is the one we have in the Department of Anatomy in the NPTRU, so with which a number of feet have been recorded. This runs basically on the principle of you having a heated filament going through a filament stepper, right, that adjusts the movement, then a thermometer that regulates the temperature such that you have a hot end here. This is what the filament passes through, and then coming out through the nozzle as a molten plastic, heated plastic that have been melted, which is what will then be used to form or fabricate whatever is intended. So it uses the extruder system, as you have seen. So what are some of the applications of these to Labwares? Whether you're into neuroscience or not, whenever you're conducting biomedical research generally, what are some of the applications which you, as the thinking youth, also need to start thinking about harnessing, right? So for some reason, some things like cryptocurrencies have become so popular, especially amongst the youths, and with the same amount of flexibility and thinking is dedicated to things that have to do with harnessing 3D printing into solving scientific problems. We also would make a great breakthrough, and this is going to become a more popular means of fabrication or of solving problems. If we had to ask how many are the divisions of biomedical sciences, there wouldn't be a single way to categorize them or to classify them, divide them, right? So let's just take this as a typical way of doing that, such that we have different aspects. And because we have diverse aspects of biomedical sciences, as well as diverse aspects of or diverse subspecialties of neurosciences, we are definitely going to be having diverse research questions. Our research questions will be the same. We'll have different problems we want to address within the society. So from your perspective, your research question likely can be different from mine, and likely going to be different from the next person coming from another subspecialty. Even from the same subspecialty, you tend to concentrate on different things at different times. So diverse research questions bring us together. However, there are some common denominator to whatever research that you're trying to pursue. It's always likely that you've run into needing some things that the companies won't readily make for you, right? You need some things that the companies won't readily make for you. And so you have to think of how to alternatively fabricate that yourself as a way of self-help. All right. When you find no help, you start thinking of how to help yourself. Next, it's possible that you intend to make a common technology become more flexible, such that it's able to adapt to different parameters, different conditions, and able to serve you on different levels. Once you have this intent, this is also something common to whatever subspecialty that you find yourself. It's also possible that you have a high detail of a customized design that you want to achieve, right? You need, it has so much high detail that you readily can't buy it off the shelf. You don't find it off the shelf. You don't find it fabricated already, or you have to order it from somewhere that is very far. So when you need to customize some design yourself, then uh, this is a common research denominator to whatever research question everybody has in mind. And the fourth thing that brings all this research together is that at one point or the other, you always need to make crucial instrumentation cheaper. Maybe some tool that you need is too expensive, and you need to make it cheaper and make it more accessible to people. So there's four main denominators make you a candidate for employing the concept of 3D printing. All right. And gone are the days, gone are the days when you have just one material that is so popular in the space. There are diverse materials now that can be subjected into 3D printing. Okay, they are no longer as usual. So you just think of your research question uh, and go on, right? And think of what printer serves you best and get it done. Then get it done. So you could have metal, chocolates, can companies now are able to companies that are able to design different geometries of chocolates that you consume. A lot of them now employ 3D printers, all right, such that the kind of printer that can handle the chocolate, the ingredients, the raw materials of making chocolate. So you need bio ink printing materials for you to be able to print living tissues, right? To be able to print living tissues that are applicable in medicine. So it is known that recently engineers at Harvard developed a novel combination of bioinks that can be used in tissues and mimic human tissues. That's what you find. All right. And also you're able to print materials that are as close to bone as possible, such that they can even be used, with titanium implants, and they serve the purposes that bones will do. So all those implants are materials that are 3D printed. Then sandstones as well. At times, what you want to make is of glass material. Glass can be 3D printed. Medication as well, used in pharmacy, can be 3D printed. The skin grafts, all right, also have potentials of being 3D printed, as is obtainable already. So you will agree that it's limitless, essentially. It's limitless. So it's just like the silicone parts that are printed, ear aids that are also made of silicone. So all of these go to show that the sky is the limit. And if you like to see it as a starting point, all the best to you. So there is hardly a material, and it's evolving. You keep having new ones each time. And the good news as well is you could think of recycling, right? You could think of those waste material you have in your community. There is fortunately a group now that is working hard to ensure that, I mean, 3D printers, 3D printers are created based on the material that you have access to. Whatever raw material is so much is available in abundance in your area, in your locality, uh, this company is trying to be able to develop a printer, a 3D printer that will run on that. So what determines the kind of printer you have is what kind of material you want your products to be made of, all right? And then that determines what kind of material you must use, and that determines what kind of printer runs on that material. So if you want metal material, you definitely need an advanced 3D printer that will be able to print metal. Will be able to melt the metal, reconfigure the metal, print metal, and so on. So it means that it's an evolving science. And while it's a wide, it creates a kind of spectrum such that there is really no end in sight. It's evolving, it's changing. That's a good news. So this has been applied in stem cells, and these are things I can give you some time to ruminate about. Stem cells are so important that they are more or less the body's raw material in terms of their pluripotent potential, totipotent, pluripotent potential. So once you're able to isolate them and then be able to reproduce them, okay, in vitro, definitely achieving that will help to understand human cell function much better. And it becomes a highly valuable concept in addressing regeneration or or, or disorders that are based on neurodegeneration for you to be able to regenerate neurons, and also in trauma. So how in 3D printing neural stem cells, I mean, printing 3D printing stem cells tells you that you wouldn't be using the same printer that you use for plastic, right? It has to be something that is under as much of sterilization as possible. So assembly of neural 3D networks is the first thing to achieve. You first of all establish 3D networks and then you have a well-defined duplicatable scaffold, 3D scaffold for those stem cells that will be capable of imitating, if you are talking about neural stem cells, so that will be able to imitate the brain activity, the network in the brain, the network of neurons in the brain. So that's why assembly of those neural 3D networks is the first thing. Thereafter, you create a scaffold that will be reproducible. Each time you want to reprint things that have to do with that network, then you test the modulatory effects of pharmacological and toxicological compounds of interest that you have. So there are about 5 approaches to 3D printing stem cells today. You can use dye-based layering, such that you employ coloration in differentiating blood vessels or capillaries, from the other, albeit artificial, but they are as close to bionic bionic tissues as possible. Another way is to flash freeze some 2D layers that have been printed. You flash freeze them into 3D. That is, you print each layer, each 2D layer, until you are done printing. Then you flash freeze each successive 2D layer of the entire bionic material, bionic stem cells, into 3D. Right. Another way is to populate a 3D printed cellulose. Cellulose is more like the material used in making paper, right? So the cellulose is that makes the scaffold, the structure, skeletal structure of the organ being targeted, the organ being, the organ of interest. You first of all make a cellulose of that entire structure before now populating it with 3D with the cells, with the stem cells. So you first of all create the scaffold, more like you creating a balloon before now injecting air into it. So you first of all 3D print the structure of that entire thing before now populating it with the stem cells of it. Another way is to corral the cell placement using ultrasound. The stem cells, you are 3D printing it by corralling. All right, corralling the placement of those cells using ultrasound. And the fifth method used and employed in 3D printing for stem cells is to integrate live cells into bionic prosthetics. First of all, create the bionic prosthetics before now injecting stem cells into them. So this tells you how much potential lies in 3D printing for stem cells, as important as it is, right? Then going more into physiology, there is this, looking at this is a flexi drive. Flexi drive. At times you want to make recording of what activities that are going on in the brain of animals. So here is the flexi drive itself. This is the schema of what it contains inside. It has 16 channels, connector, the drive body here. All of these are springs. All of these are springs. And this is what it looks like on the external. It is placed on the animal's head. What is the purpose? It sends electrodes into the brain of the animal while the animal is alive and moving, all right, in order to record electrical signals that tells us. So at the end of the day, the signals that are picked come out in graphs like what you have on this uh, down right corner. So this is the flexi drive, and it's something that is 3D printed. This is particularly from the works of Wojciech in 13, and it describes the flex drive as a spring-based drive mechanism that reduces implant weight and, and complexity. So this animal, despite all of these things that have been built into this 3D printed uh, body of things, it is not longer than 22 millimeter from head to cap, right? Not longer than 22 millimeter, and the total of 2 grams of weight. So whether you are putting this on mouse or rat, a rat typically weighs about 150 grams, and this structure is weighing just 2 grams. Even a mouse is weighing on average 25 to 30 grams. So because you want to record the electrical signals in them. So this makes the weight bearable for the animal. And the spring-based mechanism makes you not having to, shift or reinsert, remove and reinsert. Okay, so the animal is able to move with it, not feeling the weight of the material as it moves. So this is a device used for optical control and highly parallel chronic recording of neuronal ensembles in freely moving mice. And this has a lot of advantages beyond some that have been made earlier, in the sense that in this particular version, it is ultra light, lighter. You can see it's evolving. So it's lighter, not as heavy as previous versions. These, unlike the 4 to 8 channels or electrodes that are in previous ones, it has 16 electrodes and records up to 64 channels and doesn't weigh more than 2 grams. This is an application of 3D printing through electrophysiology. And then these are further customized implants. These are cranial implants that are adapted for use in primates. And the selling point of this is, of course, it's made of titanium, so it is printed with a titanium printer. It is tailored towards the anatomy of individual animal. So you want to know the surface anatomy and the curvature, the contours that you have on the skull that will make the cranial implant fit much better. So for primate electrophysiology. Yeah, first of all, it was printed with plastic for prototype. So a plastic 3D printer was uh, used to prototype it before the final one was now made in titanium. So also, this is an ethoscope. An ethoscope is used to record the behavior of flies, an ethoscope. So here you have it, and you have the flies within vials. Vials meaning transparent bottles. Flies like Drosophila melanogaster, all right, they are being recorded, the behavior are being recorded within these vials that have been laid down here. And this ethoscope with its different parts is what is used to observe them. You can see a camera here. Camera is right here. So that is used to observe the behavior of the animal. This is also 3D printing. Okay, and you can read more about these. So it is actually 5 to 8 times very expensive than the commercial one. If you 3D print yours, so long as you understand the mechanism and you're able to design the structure around it. So you design the structure, you design it digitally on a computer. It has 3 stages. 3D printing has 3 stages. You first of all design your material digitally. Thereafter, you convert the file, the G code, the STL file into a G code, that is slicer, and then you transfer that file into the printer, which eventually executes the printing job. So just like, think of the 3D printer like the 2D printer you use in printing papers. 3D printer is more like a fabricator, a fabricating machine, right? That makes it easier to understand. But it is a printer as well because there is a soft copy, a soft copy somewhere that you now use to make the hard copy. So just like you will transfer files with your USB, your flash drive into a computer, and you connect your computer to the printer to do the printing job. In this case now, you are also designing your soft copy material item in the computer. Then you take it, you convert it to the right format that will be recognizable by the printer, and then you send to the printer to do the printing job. So this is an oscilloscope. And next, just with €100, right? With €100, an entire laboratory, so to speak, where you are able to do fluorescence microscopy, optogenetics, and accurate temperature control in zebrafish Drosophila C. elegans was achieved using 3D printing. What you are looking at here is a combination of 3D printed material. The entire white materials here are 3D printed. It's a scaffold. So here you have an adjuster, a regulator that helps to adjust the stage. You have a more like a CPU chip system that we call the Raspberry. The Raspberry it does the job of a computer, right? So that's why you're not seeing a B computer here. The computer itself, the chip, that's the Raspberry Pi is giving is entrapment, and you have this source of light. You can see the Raspberry Pi camera looking down. Here is where you have the animal being observed. You have the stage, you have the light coming down here. So this entire scaffold structure that is used to house the material is 3D printed. And whatever recording is made is generated by the Raspberry Pi camera, gets sent into the Raspberry, and then you need a display system to see, to view, and that's why you see this screen here. So depending on the size of screen that you desire. So these are also some of the technologies being employed in Entrepreneurial to make recordings. So this brings 3D printing into fluorescence microscopy, because at the end of the day, you expect some of these things to fluoresce. So you can adapt it for as much of the technologies as you want. So you can use it to conduct behavioral data. So you just get these materials that can be purchased easily by a lens camera that will be attached to the right thing. But the entire scaffold that will help you to keep everything in place rather than being disjointed is the 3D printer. So this is definitely plastic, right? There's plastic, and this has brought 3D printing close to neuroscientific research. And another thing that is possible with 3D printer are labwares. Labwares, physical tools you need in the lab, physical tools you need in the lab. That includes micropipette, centrifuge, what have you, so many of them. So here is a typical centrifuge that was printed in the NPTRU, and it is also made of plastic, and it was printed using the NPTRU printer. And this was after we were able to make over 400 face shields and nose masks that are 3D printed during the COVID. And here is the publication that you can read more about. So generally, making labwares with 3D printers run on the basic principle, and that is for you to understand the working mechanism of each tool that you are trying to make. Here is an incubator. The job of an incubator is to incubate, to help you to deliver some level of temperature towards whatever it is you're trying to incubate. So you start thinking about alternatives, and eventually you will be able to adapt and modify and refine into something that will be reproducible all the time for your research, right? The work of a centrifuge, as shown here, is to spin. So you start by thinking of what spins, what are those things that spin around that you can start adapting into achieving what you have achieved. Just like you saw in the 3D printed microcentrifuge, this is the principle it runs on. So this is a PCR machine. It delivers it delivers level of temperature, heat, or cold, to the DNA fragment that is intended to be replicated, to be amplified. So You start by thinking of how to put some battery parts together and what have you, water, and then how to micro increase the temperature and reduce it. And eventually something is going to come out. So it is to, first of all, not see gadgets as untouchable things. You'll be ready to understand the principle with which they work, and that's the only way you can try replicating what they do. Over there is the horizontal gel electrophoresis tool apparatus. And if you think of what it does, it's more like to shock to deliver some level of electric current. So when you put some things like this together, some of these electrodes together, you start appreciating how to make a headway where you don't have it. So when you eventually think of how to save the situation, the concept of reproducibility becomes important in science, because when you make a big breakthrough, another person is going to be able to follow that protocol, do the same thing, and get a similar result, all right? If not the same result. So if you do it in your way, and I want to think about reproducing, following your footsteps, and I end up doing it in another way, we definitely won't be getting reproducible results. And reproducibility is very important in science, such that if somebody wants to get all the spare parts that makes a computer, you can imagine how bogus it's going to look like, the computer is going to look like at the end of the day, with disjointed things everywhere. But to curb, to curb that and to address that is why we need 3D printing as a means of achieving reproducibility. So this is one of the last examples we're doing. This is a two-photon microscope. You can imagine how many thousands of dollars and hence how many millions of naira these will cost, all right? And this is the typical standardized commercial one, right? And if someone were to think of, okay, creating something like that, bringing different subparts, because complicated is typically several simple items that you bring together that make up whatever you consider complicated. Every complicated machine is made up of different simple parts, only that those simple parts are so many, all right? So if you try to reproduce a two-photon microscope, what you have here as a two-photon microscope, trying to reproduce it, you bring different parts together. Here is a typical paper tape, right? Here are lead strips you have all around here. And here you have a potty. Here you have soda can, typical soda can, the Coca-Cola, you know. It's all serving a purpose, right? Here you have a phone charger around here, and then you have microcontrollers all around here. You'll see that it's quite complicated, so it becomes difficult to reproduce. But as we tag along, you eventually will come to that meeting point where you achieve an alternative that is also going to be what? Reproducible. And that happened not too long ago in 2018, where Delmans and Hasselhoff were able to bring together these U-Cube system parts library such that you are able to assemble them yourself. So it comes in a pack, and you as a researcher is able to get this pack. In fact, if you have a metal 3D printer, you can also print the parts yourself. The protocols are there online. They are off the shelf for you to harness. And then you have something like this. You can imagine, you can see how it is well constructed, neatly measured and assembled. So these are some of the things you are able to do with reproducibility, having harnessed 3D printing. So you are able to easily adjust the dimension to what you are trying to achieve and what you are trying to investigate as well. So for this, metal is, of course, the material of choice. So it begs the question for you, it brings you into under the spot, starting to think. And you have to think right. You have to start asking yourself as a researcher, what's one thing, what's two things, what three things do you consider to be worthy of 3D printing in your research project? What are those things that you consider worthy of printing in your research project? You need to start thinking right, and you need to get it right, because at times some things are probably not worth 3D printing, right? Look around you, look at all of those items that are needed in the laboratories, and start thinking of what principles they run on and how you will harness them into achieving something, into achieving a viable alternative. So what are those aspects of them that can be 3D printed? What are those aspects of them that 3D printing can help with? What are those aspects of them that can be reproduced and then make it more accessible to people, make it simpler to operate, make it easy for people to read, make it easy for people to reproduce with less cost. So you definitely have to be thinking big because Africa needs your brain. So we agree that we haven't got the money to match up with the global north, but something that we need to do apparently is to rely on open source hardware. These are things that you create, you fabricate, you generate, and share the ideas to the scientific community because everybody is ready to acknowledge you. So it's not something you do and you keep to yourself. Really, these are things that are needed. I mean, perceptions of open source are things that are needed now in scientific community in order for us to advance as a country. I mean, in order for Africa to advance really as a continent. So it's important. Yeah. And when you make them open source, you probably beg the question of why. Definitely, despite being open source, you are able to help in in providing services with them. So if you are able to generate things like this and you're able to provide them to laboratories, definitely you have a long entrepreneurial potential to accompany. So open source hardware serves as an alternative the money. When we don't have the money, open source hardware comes as an alternative way of achieving that, and then you add levels of creativity to it. So I like to usually conclude on this notion, on this talk, where Mandela says that education is the most powerful weapon with which you can use, with which you can change the world, right? So one thing we'll just add to that, while education is important, you also need that readiness to help yourself, right? The readiness of self-help is also important for you to add to the education in order to do well to changing your world. Meanwhile, taking for thinking, there are some times that you need to be so sure that it's the right thing, it's the right tool to employ for yourself. So when are those times you need to use 3D printing? Yes, to all of what we have said about 3D printing, but careful, be sure that the quantity you need to make is perhaps relatively low. You need to use something between 1 to 10 to 50 copies of those materials you're trying to 3D print, right? It still falls within the range of what you should 3D print. But when you start getting beyond that, you need hundreds of those materials. You probably need to start exploring other alternatives, perhaps much bigger 3D printers, perhaps much bigger equipment, much bigger ways of creating this. Taking the face shield that we printed in the research unit, for example, with a single printer, we're able to create up to over 400 face shields and the nose mask with a single 3D printer. Why did we do that? Because this was a time when there was lockdown. There was no importation. Talking about the early parts of 2020, right? Between March and June, March and April, March, June, July. There are times that even commercial companies haven't gotten their footing. There was no room for importation of anything. All right. This was a time where it costed gold to get nose mask, face shield. Face shield wearing even so common. So we had to swing into action and employ this, mitigate the scarcity in the community. And it really helped because of the beneficiaries were the teaching hospital that was next to the university. So we wouldn't have done that if there were commercially viable alternatives. So even the commercial people were still trying to get their footing. There was no room for that yet. There was lockdown everywhere. Even if you wanted to go out to get, you couldn't go anywhere. Even if you wanted to bring it in the country, you couldn't come into the country. So that was why we had to bend over backwards and get that executed. So when the alternatives came, it became clear that there was no point in expending so much, all right, of filaments in making things like that will be gotten with. So you also think around things that you can achieve with wood, things you can achieve with plastic, or things that are already available for sale. You don't need to go through the stress of reinventing the wheel or printing 3D printing it. You could get it for something cheaper. So be sure that what you are trying to achieve is perhaps a complex geometrical design that is critical to the parts function, and you don't find that. So this you need to take note of so you don't start killing like this sledgehammer, right? especially when you even miss. Usually you don't get to, usually you get to still miss because you are using too big a tool in achieving something that is trivial. So at times, you probably don't need to use a 3D printer when those things are readily available. But when you are trying to fabricate something that is quite peculiar, unique, then you should consider 3D printing. So you should be sure that you are aligning technology's peculiarities, different types of peculiarities, align those peculiarities to your most important requirements in the research. So that way, you will have harnessed potentials, and you will have, perhaps you need some parts that with the print, you make this from metal. A part of it needs to be metallic, another part needs to be plastic, another part needs to be glass. Perhaps you can exploit all of that and bring them all together into one. So why the wait? Just for you to assess your need, your research need effectively. Imagine your solution. Just imagine what the solution should look like. Then you clarify your goals. What exactly do I want to achieve? And then what tool do I need to achieve it? What now do I want to print? The 3D printed tool material needs to look like this have this component, should have this configuration. So you need to clarify all of that as well. You need to show all of that as well. So you clarify your goals, and then you determine what material you need. Should you run on plastic? Should you run on metal? Should you run on glass? Should you go for bionic materials in case you're trying to print yeah, perhaps you want to print some anatomical models as well. So you need to determine your desired materials. You need to be sure what materials you need, and you acquire a reliable and non-technical printing method or machine. So FDM is usually more affordable and easy to operate for starters, and it's always ideal for beginners. And you get printers like Makeup Boards, Ultimaker, or Creality Ender series that are much cheaper, than others. So FDM models are safer for beginners. They get a design that is sound and grounded in the limitations of the 3D printing process. Then you're good to go. Good to go. So if you are asked the question that is it relevant, is 3D printing relevant to neuroscience or to biomedical sciences? The answer is definitely a big yes. So because it has a far-reaching amount of possibilities and vast neuroscience potential. So before we conclude this talk, it's not all about 3D printing, just adding another dimension to what you can think of. Some of you have a flair for art, Make no mistake, what you're looking at here are plates, utensils, plates, right? More or less like ceramic plates that you know. And if you haven't forgotten, this plate is bearing the histology of a testicle. Here is the histology of the liver. Here is the histology of the kidney, the pancreas, the bone, the esophagus. Here is the thyroid gland. And here is the back of the plate for you to be sure, right? Who thinks of things like this? Ooh, so this is another dimension, arts. We're talking about histology. We started out talking about histology or histological materials, and now we are bringing arts into it. So those for the micrographs, who is meant to understand them better than the histologist that you are, the anatomist, right? Who is meant to understand them, who is meant to appreciate them. So this is what we can call histological arts, if you agree with me. Here are the same plates again. I'm sure you really desire to have one. Histological art. So meet the anatomical artist. This was the lady that made this thing. Her name is Emily Evans. You may like to read more about her. She had a degree in anatomy and cell biology, not in photography, right? So she identifies as a freelance medical illustrator. And I'm sure that's something some of you may be interested in pursuing. You can read more about her in this reference that I have. So it adds to that understanding that and self-help and creativity is highly necessary. These are references you can consider again. You can consult it again to have further read on what we have discussed. And I like to conclude again. Scientific research is one of the most exciting and rewarding occupations. I appreciate, I appreciate these bodies, these organizations for the amount of impact and influence that they have had on my research exposure. and as well as, with grants, travel grants here and there. These I am grateful to all these bodies, the International Trust, the International Research Society, International Society for Neurochemistry. All of these bodies I'm a member of, and lots more. And I can't but appreciate those that have been the working force of the Neuropsychiatry Research Lab of through transgenerationally of different sets. So I appreciate every one of them. Those are my research students. So I'm happy to receive your questions anytime through any of these channels. Uh, yeah, so feel free to send me your questions. I hope you have some fun with the lectures, and I wish you all the very, very best. So I also look forward to having your positive feedbacks, comment sections, about what ideas you have, so we can perhaps rub minds if you need some assistance, and so that we can start doing greater things, okay? Or things that you think you will be able to achieve with ideas that have been discussed. Wishing you guys all the best. Have a very nice rest of the day. Bye.